Today we will discuss about cell division. As we all know that growth is the important character of all living organisms. This is the reason why cell division becomes very important for all living creatures. All cells reproduce by dividing into two daughter cells. Cell division is essential for growth, repair and reproduction. Many cell cycles transform a single cell to a multicellular organism. Phases of cell cycle are interphase or growth phase and M phase or mitotic phase or division phase. Interphase is the time lapse between two successive M phases of cell cycle. The cell prepares for division, grows and DNA replication takes place. Interphase is further divided into three phases G1, S, G2. G1 phase or gap 1, it is the gap between previous mitosis and DNA replication of the next cell cycle. In this phase, cytoplasmic growth occurs. Mitochondria and chloroplasts are duplicated. S phase or synthesis. In this phase, DNA replication takes place. The DNA content of the cell doubles. It is important to note that the chromosome number remains the same. G2 phase or gap 2, cell continues to grow and prepare for mitosis. Protein synthesis takes place. In an adult human being, there are many cells which do not divide, e.g. heart cells divide occasionally only to replace injured and dead cells. These cells enter an inactive phase called G0 or quiescent stage of the cell cycle. M phase, this is the phase where actual cell division occurs. The number of chromosomes in the parent and daughter cells remains the same, so it is also known as equational division. M phase consists of karyokinesis, nuclear division, and cytokinesis, cytoplasmic division. Karyokinesis or nuclear division is followed by cytokinesis, i.e., division of the cytoplasm to give rise to two daughter cells. Karyokinesis occurs in four sequential stages: prophase, metaphase, anaphase, telophase. Karyokinesis is followed by cytokinesis. In prophase, chromatin fibers untangle and condense to form chromosomes with two chromatids attached to the centromere can be seen clearly. Each of the duplicated centrosomes radiates microtubulous asters. Mitotic apparatus constitutes spindle fibers and asters. Golgi bodies, nucleolus, endoplasmic reticulum and nuclear membrane disappear. Prophase is the longest phase of division stage. Prophase is followed by metaphase. In this phase, two sister chromatids attached by the centromere aligned at the equator, i.e. metaphase plate. Each chromatid is attached to spindle fibers from opposite poles at kinetochorus. Metaphase is followed by anaphase, in which splitting of centromere and two sister chromatids separate and move towards the opposite poles. Sister chromatids now become the daughter chromosomes. Anaphase is the shortest phase of division stage. Anaphase is followed by telophase, that is terminal phase of karyokinesis. Events in telophase are reverse of prophase. Chromosomes cluster at opposite poles and decondense to form chromatin fibrous. Nuclear envelope develops around each cluster of chromosomes and two daughter nuclei are formed. The nucleolus, endoplasmic reticulum and Golgi apparatus are reformed. Cytokinesis, separation of cytoplasm, takes place after two nuclei are formed. Cell organelles get distributed between daughter cells. In animal cells, cytokinesis starts at periphery in equatorial plane and moves towards center in centripetal node. In plant cells, cytokinesis starts by cell plate formation at center in equatorial plane and moves towards periphery in centrifugal mode.